Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show.
Keratinization. It is also known as cornification. It is a process of keratinocyte differentiation into various suprabasal layers, ultimately resulting in the formation of a keratin-rich permeability barrier, that is stratum corneum. It includes molecular events such as formation of keratin and stacking of keratin intermediate filaments, which is mediated by filaggrin, filament aggregating protein, and formation of a cornified cell envelope. What are keratins? Keratins are intermediate filaments in the cytoplasm which forms cytoskeleton of keratinocytes. These keratin intermediate filaments not only forms cytoskeleton of keratinocyte, it also links to desmosomes at one end and dermoepidermal junction, that is hemidesmosomes at the other end in basal keratinocyte. Here you can see keratin intermediate filaments are also known as stonofilaments attached to desmosomes and hemidesmosomes. There are total 54 human keratins classified based on pKi, that is based on pH. One is acidic, another one is basic. Based on its contents and consistency, one is soft or epithelial keratin found in skin, which is mainly containing glycine and serine, which is low in sulfur and disulfide bonds. That's why our skin is very soft. Hard or trichocyte keratin found in hair and nail, mainly contains cysteine and proline, which is rich in cysteine is rich in sulfur and disulfide bonds, which provide mechanical resilience to the hair and nails. These keratins forms obligate heterodimers. That means one acidic keratin monomer binds to basic keratin monomer, forming a heterodimer only, ultimately leading to a filament. Coming to its applied aspects. Keratin has varied expressions according to the location. In epidermis, in basal layer, cells express keratin 514 heterodimer. If it is defective, it leads to the genodermatosis called epidermolysis bullosa simplex. In rest of all the layers, suprabasal layers, keratin expressed is 110. If it is defective, it leads to epidermolytic ichthyosis. In palms and soles, keratinopathies include palmoplantar keratoderma. In primary ridges, keratin 19 is expressed. Its defect leads to epidermolytic PPK. In secondary ridges, keratin 6A 16, 6B 17, which if defective leads to pachyonychia congenita. In hair, there are multiple keratins. Here 81, 83, 86. Defect leads to monolatrix. Epidermopoiesis means production of epidermis is by keratinocyte stem cells present in basal layer, which gives rise to all the layers of the epidermis. This epidermopoiesis is regulated by balance between stimulatory and inhibitory signals. These proliferative cells, that are keratinocyte stem cells, enter cell cycle, which is having 4 phases. M phase, that is mitosis phase, where a parent keratinocyte divides into 2 daughter keratinocytes. Next, after mitosis, post-mitotic G1 phase, that is G means growth phase. Majority of the growth occurs in this phase only. Next, S phase, that is DNA synthesis phase. And then G2 phase. Here also G means growth. It is occurring before mitosis, that's why pre-mitotic growth phase. Here any defective DNA is there, will get repaired in this phase. Ultimately, mitosis occurs and 2 daughter cells will form. It has 3 options. One is continuing the cycle as it is. Next one is taking a break and entering quiescent phase, that is Q0 phase, whenever a signal comes, it can again continue its life cycle by maintaining the potency to divide. And then majority of the cells will decide to take third option, that is terminal differentiation. It won't enter cell cycle, it won't enter quiescent phase, it will become non-mitotic and undergoes terminal differentiation. Majority of the post-mitotic cells become non-mitotic and lose integrin expression. Integrins are the one transmembrane proteins which attaches basal cell to the basement membrane zone. Now it gets detached from the basement membrane zone and moves towards the skin surface to form stratum corneum. During this movement into various layers of the epidermis, they undergo a process of terminal differentiation, that is nothing but keratinization. First, from basal layer, because of losing integrin expression, it enters the spinous layer. Here cells will produce keratin 110 instead of keratin 514. It is the first step in differentiation. And calcium levels will be increased in each layer. Because of this increased calcium, upper layers of spinous layer will start producing woodland bodies. And then it will enter granular layer. There along with the woodland bodies, also known as lamellar granules, they produce keratohyalin granules also. These keratohyalin granules contain mainly proteins which contribute to the formation of cornified cell envelope. Lamellar granules contribute to the formation of cornified lipid envelope. After reaching stratum corneum, these cells already have two types of granules, keratohyalin granules, lamellar granules. Lamellar granules, before reaching the stratum corneum, at the junction of granular layer and corneal layer, it will release lipids into intercellular space, forming cornified lipid envelope. Now, at the level of stratum corneum, cells will form highly insoluble cornified cell envelope of proteins cross-linked by transglutaminase 1. We have already seen keratohyalin granules contributing to cornified cell envelope. Same proteins along with desmosomal proteins, keratins, all together form a cross-linked network of envelope with the help of transglutaminase 1. If the transglutaminase 1 is defective, it will lead to lamellar ichthyosis, where a cornified cell envelope will be seen deep to the plasma membrane of a corneocyte. Thus, ultimately, the final product of keratinization is a dead cell, which is shed and replaced by new ones continuously. Here ends the journey of a keratinocyte from taking its birth through a keratinocyte stem cell at stratum basal layer, traveling all the layers and ultimately reaching a dead layer, stratum corneum, where the cell will lose all its water, organelles, nuclei and becomes a corneocyte, which is nothing but a dead cell, which will have cornified cell envelope. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.